Hello, my dear student friends. In this module, we will talk about the measurement of bacterial growth. We will talk about the different methods which are used to count the number of cells, the number of colonies, and also the turbidometric measures to measure the cell density.
Is the measurement of bacterial growth. There are a number of processes carried out by microorganisms. These processes include fermentation, degradation, spoilage, etc. While it is very essential to determine the type of microbe carrying out these reactions, it is equally essential to know the number of microbes carrying out these reactions. As part of daily routine, the laboratory microbiologist often has to determine the number of bacteria in a given sample as well as compare the amount of bacterial growth under various conditions. Enumeration of microorganisms is especially important in dairy industry, food processing plants, and water treatment plants. Many food processing plants measure the level and type of microorganisms present in their food by doing counts on selective medium. In addition, viable cell counts are performed when optimizing heat treatments for food processing. Sewage treatment plants routinely sample and count the microbes present in their treatment systems to ensure the correct type and numbers of bacteria present. Also, coliforms are enumerated in water to determine the potability of water. There are many methods to measure bacterial growth, each having its own advantage and disadvantage. The method you use would depend upon the organism in question and the experimental conditions. These different methods include measurement of cell number, measurement of cell density, measurement of cell mass, and measurement of cell activity. Let us first move on to the measurement of cell number. These methods include direct counts and indirect viable cell count. The first method is direct microscopic count. One of the simplest methods of determining the number of microorganisms is by direct counting. In this method, a counting chamber consisting of a ruled slide and a cover slip is employed. It is constructed in such a manner that the space between the cover slip and ruled lines on slide can hold a small and known volume of the sample. There are 16 small squares in the large double-lined squares that are actually counted, making the volume of a large double-lined square 1 upon 1,250,000 cubic centimeters. The normal procedure is to count the number of bacteria in 5 large double-lined squares and divide by 5 to get the average number of bacteria per large square. If the bacteria are diluted, such as by mixing the bacteria with dye before being placed in counting chamber, then this dilution must also be considered in the final calculations. The formula that is used for direct microscopic count is the number of bacteria per cc is equal to average number of bacteria per large double-lined square into the dilution factor of the large square. Though this method is the simplest, there are some disadvantages that are associated with this method. They are using a dye for visualizing the cells often kills them. Thus, it is not possible to distinguish the dead cells from living, and hence it gives us the total cell count rather than the viable count. Second, a phase contrast microscope is necessary for unstained preparations. Third, small cells might be missed during counting and hence leading to errors. And fourth, accuracy can be achieved only by experience. The next method of measurement of cell number is the plate count, also called the viable cell count. Most of the times, we are interested in counting only living cells, and for this purpose, the viable cell count is desired. A viable cell is a living cell which is capable of cell division. The viable count can be determined by allowing a small volume of sample to grow on a suitable agar medium and form colonies. It is assumed that each viable cell yields one colony and the number of colonies is counted. There are two methods which are routinely used for this purpose: pour plate method and spread plate method. The success of both the methods depends on the proper dilution of the initial sample. Usually, more than one dilutions are made and it is therefore appropriately called the serial dilution. 10-fold dilution tubes are made. For this, 9 ml distilled water or physiological saline is taken in the tubes and the tubes are labeled as 10 to the power minus 1, 10 to the power minus 2, 10 to the power minus 3 and so on. As you can see in the figure, 1 ml of the original sample is added in the first dilution tube to get 10 to the power minus 1 dilution. The contents are mixed thoroughly and 1 ml from 10 to the power minus 1 dilution is added to the second dilution tube to get 10 to the power minus 2 dilution. This process is repeated till the last dilution tube is reached. In pour plate method, a known volume of sample is poured into a sterile Petri plate as can be seen in the figure. Sterile molten agar is poured into the plate and the plate is gently rotated so as to ensure proper mixing of the sample and agar. Alternatively, the sample can be added into a sterile molten agar tube, properly mixed and then poured into the sterile Petri plate. In spread plate method, the sample is suitably diluted and then 0.1 ml of the sample is spread over the surface of the sterile agar plate with the help of a sterile glass spreader as shown in the figure. The surface of the agar should be dry so that it soaks in the sample. It is not desirable to use a sample volume more than 0.1 ml because the excess volume does not soak in and causes the colonies to mix and fuse so that it becomes difficult to count them and also leads to errors. Finally, the plates are incubated for 24 hours and the colonies are counted. In the case of pour plate method, the colonies grow in and on the medium, but in the spread plate methods, colonies grow only on the surface of the medium. After incubation, the number of colonies on the plates are counted using a colony counter. Generally, one wants to determine the number of colony forming units per ml of the sample. To find this, the number of colonies on a plate having 30 to 300 colonies is selected. This is because this range is considered statistically significant. If there are less than 30 colonies on the plate, small errors in dilution technique or the presence of a few contaminants will have a drastic effect on the final count. Likewise, if there are more than 300 colonies on the plate, there will be poor isolation and colonies will have grown together. The number of colonies is multiplied by the number of times the original bacterial sample was diluted. For example, if a plate containing 1:10 to the power 4 dilution of the original ml of the sample shows 32 colonies, then 32 represents the number of CFUs present in the original ml. Therefore, the number of CFUs per ml in the original sample is found by the formula number of colony forming units per ml of the sample is equal to number of colonies into dilution factor of the plate counted upon volume of the sample plated. Thus, we get 32 into 10,000, which gives us 3.2 into 10 to the power 5 colony forming units per ml. The next method is measurement of cell density. When you prepare a cell liquid medium like nutrient broth and inoculate it with a bacterial culture, say E. coli, and incubate it, you observe that the medium which was clear previously has now become very turbid. The turbidity is actually an indication of bacterial growth. The more turbid the medium, more growth in the medium. Why has the medium become turbid? It is so because the bacterial culture acts like a colloidal suspension which blocks and reflects light passing through the culture. Within limits, the light absorbed by the bacterial suspension will be directly proportional to the concentration of the cells in the culture. By measuring the light absorbed by a bacterial suspension, one can estimate and compare the number of bacteria present. The instrument used to measure turbidity is a colorimeter or a spectrophotometer. It consists of a light source, a filter which allows only a single wavelength of light to pass through, the sample tube containing the bacterial suspension, and a photocell that compares the amount of light coming through the tubes with the total light entering the tube. As the cell number increases, the absorbance also increases. When measuring growth of a culture, the term optical density or OD is normally used to more correctly represent the light scattering that occurs. This is an indirect method of measuring cell number, but still it is very commonly used because it is rapid and simple. The next type of methods are that of the measurement of the cell mass. As the culture grows, the cell mass increases. Hence, increase in the cell mass can be used to measure growth. The simplest method for this is to measure the weight of the cells. This is done by taking the samples of the culture at regular intervals, centrifuging them to get a pellet at the bottom. This cell pellet is then washed to remove any impurities and dried in the oven to remove all water. This leaves the dry cell mass, which is then weighed. This method counts both dead as well as living cells. Also, the bacterial culture has to be very dense to give you a substantial cell pellet. This method is better for those bacteria which are filamentous, like the Streptomyces species and the molds, whose growth cannot be measured by standard methods like viable cell count or turbidometric methods. This is because they form clumps when they grow. Some bacteria that excrete polysaccharides will have a much higher dry weight per cell when growing on high sugar levels than on low. Thus, the weight of the cells will differ under different growth conditions. Also, this method is tedious and time consuming. Yet, with all its shortcomings, this method is routinely used for the measurement of growth due to its simplicity and ease. The next type of methods are those of measurement of cell constituents or cell activity. Sometimes it is practically impossible to measure either the cell number or the cell mass, particularly with organisms which form a mycelial mass or form pellets or clumps. In these cases, it is better to follow the change in the amount of cellular components instead of the entire cell mass of the cell. This can be done by determining the amount of particular cell component which increases proportionately with the increase in the cell number, like that of protein, DNA, RNA or nitrogen. Total nitrogen determination is done by Geldahl's method. Since about 16% of protein is always nitrogen, Geldahl values can be converted to get protein values. Alternately, the protein can be determined by any standard colorimetric method. The following CO-calcio method is routinely used to estimate proteins. Also, Production of carbon dioxide during fermentation can be measured by using the apparatus which you can see on your screen.